Hello, my name is Simon Liu. I am a bioinformatics assistant from the Chao Zhang Lab in the Computational Biomedicine Department in Data Science Core at BU. The title of my poster is Rogue Blood and Bone Marrow JAK2 B617F Megakaryocytes as revealed by single-cell RNA sequencing. This project is in collaboration with Dr. Katja Ravid and her team. So, starting off with some background information, the focus of this project is megakaryocytes, which are platelet progenitor cells that play a central role in hemostasis and thrombosis. These megakaryocytes are differentiated from the hematopoietic stem cells that exist within the red bone marrow. So, in this project, we are particularly interested in the JAK2 V617F mutation in hematopoietic stem cells that leads to an abnormal stem cell differentiation and development of megakaryocytes, causing an increase in the overall number of megakaryocytes and subsequent downstream platelet production. This JAK2 mutation and differentiation cascade has been found to be associated with a number of different myeloproliferative neoplasms, also known as blood cancers, as well as cardiopulmonary complications due to the excess number of platelets now within the bloodstream. So in recent years, megakaryocytes have been found in not only the bone marrow, but also in the lung and blood circulation. And recent findings from the Ravid lab have identified an increased number in the overall number of megakaryocytes in the lung and blood of mice carrying the human JAK2 mutation in hematopoietic stem cells. Individuals with the JAK2 mutation have been found to have a much higher risk of thrombosis and pulmonary hypertension. Uh, which has led to the hypothesis that the mutation of the JAK2 gene reprograms these circulating blood cells towards a possibly more pathogenic phenotype. So, in order to investigate this, an experimental model was designed to capture megakaryocytes for downstream analysis. Blood was collected from 12 weeks old male mice, and samples were stained for CD41 positive megakaryocytes. And also lineage exclusion was performed by staining for B220 for lymphocytes, TER119 for red blood cells, and GR1 for granulocytes. Flow cytometry analysis was performed to sort and gate for only CD41 positive megakaryocyte cells while excluding all other lineages of the hematopoietic stem cells. So with that, 10x genomics chromium single-cell RNA sequencing was performed on the megakaryocytes of JAK2 mutated and wild-type control mouse blood and bone marrow samples. And traditional pre-processing and subsequent downstream analysis methods were utilized using SURAT and R to dimensionally reduce, cluster, and perform differential gene expression analysis. With those differentially expressed genes, the database for annotation, visualization, and integrated discovery, also known as DAVID functional annotation tool, was used to identify enriched pathways and functions specific to the JAK2 mutation. So moving to some preliminary results of single-cell RNA sequencing data, we have here the dimensional reduction UMAP plots. For 1A, the blood JAK2 mutated compared to the blood wild type, and in 1B, we have bone marrow JAK2 mutated compared to bone marrow wild type. And while there is some overlap between the megakaryocytes of JAK2 and wild type, we can see that there is a transcriptionally differentiation of certain megakaryocytes that's unique to only the JAK2 mutated condition for both blood and bone marrow. And this shows some additional evidence to support our hypothesis that the JAK2 mutation is indeed causing abnormal differentiation and development of megakaryocytes. So, to investigate this further, we first had to validate that these are megakaryocytes that we are looking at, and we did that as shown in plot 2A, which is a violin plot of the expression of five very common megakaryocyte marker genes. So we have PF4, ITGA2B, GP5, GP6, and MPL, which are all shown to be enriched and expressed in all four of our conditions, blood JAK2 mutated, blood wild type, bone marrow JAK2 mutated, and bone marrow wild type. And with that confirmation, we were able to move forward with some differential gene expression analysis, as shown in 2B. An example of those results is a heat map of the top 50 differentially expressed genes in the bone marrow JAK2 when compared to the bone marrow wild type. As we can see on the left side of figure 2B, these genes are significantly enriched and expressed in bone marrow JAK2 mutated samples, while on the right, there is little to no expression in the bone marrow wild type. Now, taking those differentially expressed genes, we're able to feed them into the David annotational tool to get the enriched functions and pathways specific to bone marrow JAK2 mutated samples. And we were able to identify that immunogenic and inflammatory functions and pathways are enriched in the bone marrow JAK2 condition, further supporting our hypothesis of JAK2 mutation leading to a more pathogenic phenotype. So with that, we could then upscale it even further to not only include analysis of the top 50 differentially expressed genes, but include all differentially expressed genes in the JAK2 mutated condition for both blood and bone marrow. So here we have the functional and pathway enrichment results for blood and bone marrow using around 2,000 differentially expressed genes in both conditions. And we can see here that once again, immunogenic, inflammatory, and even thrombotic processes are enriched in both the blood and bone marrow in the JAK2 condition when compared to the wild type. So to conclude, from this preliminary results, we can make the claim that immunogenic, inflammatory, and thrombotic processes are all enriched in circulating blood megakaryocytes with JAK2 mutation. And this is of particular interest because in circulating blood megakaryocytes, because they are in circulation, their potential to affect other tissues directly or through secreted factors is present. So with that, our next steps is to perform further clustering, identification, and differential expression analysis of individual cell clusters representing different stages of megakaryocyte development and maturation. This analysis would allow us to determine the exact type of megakaryocytes that express these immunogenic, inflammatory, and thrombotic processes and the exact molecular mechanisms behind them. Thank you.